Hi, I'm Sapority, and today I'm going to present the study entitled as Frequency of Porcine Circovirus 3 Genome in SERA from GILTS and SOWS and their respective stillborn tissues from Spanish pig healthy farms. PCV3 was first identified in 2015 by next-generation sequencing in pigs displaying reproductive failure, especially mummified and aborted fetuses, porcine dermatitis nephropathy syndrome, and also in pigs with cardiac and multisystemic inflammation. Here we have a picture of Palinsky and collaborators of a sow. On the top we have a sow infected with PCV3 showing skin lesions, and on the bottom infected mummified fetuses. Since then, the virus has been identified in pigs affected by several disease conditions and also in healthy animals. Although all the publications of infected pigs with different clinical signs, we could see a high number of reports converging to a potential causality association of PCV3 with reproductive disease. This association is due to the viral detection in animals displaying reproductive clinical signs in absence of other pathogens, and also due the virus of PCV3 virus isolation from cases of reproductive losses. But the most significant finding was demonstrated by Arruda and collaborators, where PCV3 was found within histological lesions. And here we can see a fetal heart published by Arruda with histological lesions and also infected by PCV3. However, there is a lack of available information on the virus detection in farms with good reproductive parameters. So, the objective of this study is to establish the PCV3 infection background in normally performing pig farms. So, for that, PCV3 was investigated in 3 Spanish healthy farms A, B, and C. So, serum sample from 121 sows, being 57 primiparous and 64 multiparous, were taken at 2 time points. First at perimating, second close to ferrying. And a total of 255 fetuses from the analyzed sows were also collected at ferrying time. Lung and brain were collected from 49 mummified fetuses and 206 stillborn piglets. SERA from primiparous and multiparous, as well as lung and brain from the respective fetuses, were DNA extracted, followed by a PCV3 PCR, and when positive, the viral loads were assessed by a quantitative PCR. Statistical analyses were performed using Pearson chi-square test or Fisher's exact test in order to compare frequency of PCV3 detection in groups and farms. P-values lower than 0.05 was considered statistically significant. The samples with high viral loads at the quantitative PCR were also sequenced. 19 out of the 57 SERA from primiparous cells were positive, while all SERA from multiparous cells were negative. From the 19 positive cells, 1 was positive at perimating sampling, while the other 18 were positive at ferrying sampling. The percentage of positive fetuses from primiparous cells, which means 80%, was significantly higher than the ones from multiparous cells, which means 8%. With regards the percentage of studied tissues from the total fetuses, lung showed a higher frequency than the brain. However, this difference was not statistically significant. Here we have a table with the fetuses results. They were divided in mummified fetuses or in stillborn piglets. They were also divided by farms A, B, and C in different colors. The number of mummified fetuses positive to PCV3 was significantly higher than the number of stillborn piglets infected to the virus. With regards farms, the percentage of positive fetus from primiparous cells was significantly higher than the ones from multiparous cells, either in farms A and farm C. This comparison was not possible to perform in farm B due to the lack of collected fetuses from primiparous cells. 42 samples were selected for sequencing, based on the CT and the qPCR. However, the complete genome of only 39 out of the 42 selected samples were successfully sequenced and phylogenetically analyzed. All analyzed samples were classified as PCV3A, and the identity of them ranged from 99.2 to 100% when analyzed between each other. Here we have the phylogenetic tree constructed with samples previously classified and together with the ones retrieved in this study, which were colored by farm. So, samples from farm A were colored in red. Samples from farm B are colored in blue. Samples from farm C are colored in green. We can see that all samples from this study clustered close to each other, which emphasized the high identity between them. So, with this study, we could observe that PCV3 was only detected in primiparous and not in multiparous cells, and the virus was found in higher frequency in fetuses from primiparous than in fetuses from multiparous cells, and also in higher frequency in mummified fetuses when compared to stillborn piglets in this study. Thus, we can conclude that PCV3 can be vertically transmitted, causing intrauterine infections in absence of overt reproductive disorders. Since we could observe the PCV3 infection with normal reproductive parameters, we also wanted to check the frequency of PCV3 in cases of reproductive failure. For that, we performed another study, which I would like to briefly present to you. It's also in poster format with the reference shown here in the screen. For this study, the objective was to assess the frequency of PCV3 detection and potentially associated lesions in tissues of fetuses and stillborn piglets sampled during outbreaks of reproductive failure. The frequency of PCV3 was analyzed in 53 cases of evident reproductive problems at the last term of gestation. The samples were sent to a diagnostic service. For that, tissues of aborted fetuses and stillborn piglets were collected at two manners. First, fresh, in order to DNA extract and quantify the viral loads. Tissues were also collected fixed in formalin in order to perform the histopathology. Samples with high viral loads at the qPCR were selected for PCV3 in situ hybridization and also for genome sequencing. The presence of PIRS, PCV2 and porcine parvovirus were also assessed. PCV3 was detected by qPCR in 18 out of the 53 studied cases, where 8 showed quantifiable viral loads. 6 cases showed high loads of PCV3, which means CTs lower than 30. All 6 were sequenced and phylogenetically analyzed and classified as a PCV3A. The same 6 samples with high viral loads were tested by PCV3 in situ hybridization, where 4 resulted positive. PCV2 was found in a frequency of 9.4% in the studied cases, while Pierce virus was found in 7.5% of the cases, and porcine parvovirus was absent in all studied cases. It's important to mention that no co-infection was found in the 4 PCV3 in situ hybridization positive cases. The main histopathologic lesions found in this study were arteritis and periarteritis, usually concomitant to a relatively high amount of virus at the in situ hybridization. Here we have the histologic results on the top and the PCV3 in situ hybridization on the bottom. First, on the left, we have pictures of a splenic artery with a moderate perivascular lymphocytic infiltration, especially here, and also a tunica media swelling. While in the in situ hybridization of the same tissue, we can see the PCV3 label the smooth muscle of this splenic artery. On the right, we can see a kidney pelvis artery, also with moderate swelling here, and the PCV3 in situ hybridization result. The PCV3 genome was detected in smooth muscle and also in lamina propria of this artery. PCV3 genome can also be found in healthy tissues of the same animals where lesions were found. Here we have lung and cerebellum with no apparent histopathologic lesions. However, we can observe PCV3 genome in macrophage-like cells in lung, also in perivascular area. Also in cerebellum, we can find PCV3 genome in white matter in a high amount, and also in a moderate to low amount in gray matter. Since the simple viral detection of an endemic virus does not imply the causality of the clinical condition, the detection of PCV3 within lesions provides a stronger evidence of a putative association between the presence of PCV3 and the clinical outcome. So, these results that we presented emphasize the need to include PCV3 as a potential infectious agent to cause reproductive failure in swine. In conclusion, these studies demonstrated that PCV3 can cause intrauterine infections in absence of evident reproductive disorders, but can be occasionally detected in lesions found in reproductive failures. It's also important to mention that further and larger studies are needed to know the frequency of these findings and also the economical impact of it in the swine production chain. Thank you very much for your attention.